Good evening, everyone, and welcome to AngelMD's Oncology Pitch Club. We're very excited to have you here, and I know we've got some people who have been here many times and are experts of the whole process, but I see some new names as well. And so just want to say welcome to everyone. We've got two great companies presenting tonight, and just a few quick housekeeping things. There's a few key ways that we've set up within the Pitch Club to be able to provide input and feedback to these companies who have worked hard to not only on their businesses, but to give you guys a really informative presentation. So first, we will have live Q&A. So please do put your questions in the Q&A section. You will see there's a little Q&A right to the right of the chat button. So if you click on that, you can provide us your questions, and there's actually a feature that you can upvote. So even if you didn't write a question, if you like one of the questions and want to be sure we get an answer, you can upvote that question. And please, we invite questions from everyone, not just our oncologists. We also do have a poll. Again, we'd love to get your feedback from everyone. It just takes a couple minutes. We have a few questions about each of the companies, so we will provide the poll during the event so that you can access that and provide your feedback on their presentations. And then the last thing, which we find to be the most fun and interesting, is we do have post-event networking. And so all of our presenters will have tables after the session where you can continue the conversation and ask them additional questions and just get connected with them. So again, welcome everyone. I would like to just take a minute to hear from our sponsor, Oncology Commercial Services.
Welcome to the AngelMD Oncology Pitch Club. My name is Nathan Nagel, and I'm the CEO of Oncology Commercial Services. I bet you can't figure out what we do. Well, 10 years ago, we thought of doing lots of fancy brand names. However, we decided upon Oncology Commercial Services. And 10 years later, it seems to be going pretty well, so we stuck with it. What we do is we work with oncology companies in the UK, in the US, in the EU, and some other parts of the world are on the sanction list, so no longer. What we do is we work with any stage company and help them through the journey. What we have is a big access of oncology professionals, both in pharma and in medicine. We work both at the R&D stage, development stage, and promotion stage. How we help is with strategic advice, with tactical execution, and business development. We also have a CME license to run CME sessions. Now, what we do is we work with you. We bring in our key opinion leaders. These are helps for business development and education of the oncologists out there. So if you're based in the UK, US, or Europe, you're an oncology company, and you want commercial service support, call OCS. Thank you very much for listening. Have a good session.
Thank you to Nathan. So now I would like to turn it over to Dr. Richardson. And she is our MC for the evening. So Dr. Richardson, I'll turn it over to you.
Thanks, Jennifer. And thanks again to Nathan and Oncology Commercial Services for sponsoring our event tonight. Very exciting. And I am super excited. We have two great companies tonight that have been working very hard to be able to pitch to our audience. They spent a lot of time preparing, and I think you will enjoy both of them. So our first presenter is going to be Dr. Mari-Laure Jaspo, and she is the founder and chief scientific officer of Alacris Theranostics. Mari-Laure is joining us from Germany this evening, and I know I've seen in the chat that we have several folks that are outside of the US, which we do every time. But for our presenters, when they end up having to give a pitch in the middle of the night, I believe it's 2 a.m. there, we appreciate you being here even more.
So good afternoon. My name is Marie-Laure Jaspo, CS1 and founder of Alacris Theranostics, an innovation leader in personalized oncology. And thank you for giving us the opportunity to present our flagship product, the CMTA, which stands for Comprehensive Molecular Tumor Analysis, which is a high-precision diagnostics for cancer patients, filling a need, a crucial need in oncology. So the problem with current oncodiagnostics is that every tumor is unique, but it's unique, but it's also a complex world of phenotype, which is the result of a mesh of molecular alterations. And the problem really with the current test is that they are unable to capture this, the tumor landscape. And why is that? It's because those tests are DNA based and can identify mutations, but miss a number of events which happen in the tumor, including the expression status of DNA mutations, the gene fusion, RNA splice variants, dysregulated oncogene, prognostic gene signature, and tumor microenvironment with the immune infiltration, which all together, collectively or individually, represent targets of potential treatments. And so one way to access, the only way actually to access those events is to read out the RNA molecule, which is a functional readout of the DNA. So the issue is that by the current test, the oncologists are missing true option for personalized therapy, which might lead to ineffective treatment, drug resistance, side effect, wasted money also. So there is a gap in basically reading out what the tumor is about. So the CMTA solution that we propose is a comprehensive sequencing-based diagnostics which uncovers the tumor behavior context. And how is that? So we integrate to the DNA-seq, to the DNA information, the RNA-seq, which is a messenger information, the functional message, which give access to altered mRNA abundance, which picks up overexpressed oncogenes, splice variant, gene fusion, immune infiltration. And with this data, with this information, we use our computer programs to chart the nodes of the cancer pathways to be able to match the therapies, potential therapies, drugs to those nodes and deliver to the oncologists the CMTA report. So the benefits really for the oncologists and for the patients is to have a personalized tumor data report which provides more actionable targets, allowing to have treatment recommendation which guide clinician choice based on the tumor of the patient's data and not on guesses. So that offers new possibilities, extended possibilities for the treatments. So how that works? Basically, we extract the signal from the noise to tease out the key tumor information to get there to those to the drug targets. And so how it is that we achieve this and ahead of many companies, most companies, I shall say, it's simply because we have been working years on the issue of RNA-seq and analyzing RNA-seq. So there are 4 crucial steps. So first, we have been pioneers in protocols of single-strand RNA-seq, which provides a level of precision that we need for these diagnostics. The second step is that we have built integrative analysis, which allow to read out measures quantities of RNA-seq and integrate with the DNA information. And with that big data at the step 3, we are able to see really the important events for each individual tumor. So to have a personalized landscaping of the tumor, which is used as a basis for the drug recommendation. So that involved a number of events, as you see, which can happen and happen in general in the tumor. Some of the important ones are really the expression level of all genes. We can measure these here. You can see that, for instance, in that particular tumor, VEGF-A, which is a angiogenic factor, which is targetable, is very high compared to the mean. We can classify the tumor and read out the composition of the immune infiltration. And the step number 4 is to be able to automatically control this together in a personalized tumor report system, which will support also combination therapies, listing a number of those alterations and the matched drugs. So that is really the breakthrough here. So how does it validate? Does it really help to get more alteration and the drugs that match to it? Yes, it does. There have been several trials and translational studies which have been evaluating how this approach really boosts the possibilities to choose targeted therapy, and they all agree, including our study, that is more than 30% increase in the number of cancer patients receiving a targeted therapy guided by those type of integrating approach. So I showed an example, a case example, here on the right hand side from our study. It is a skin melanoma which didn't have a BRAF mutation. We detected 150 mutations in it, but none of those mutations were really relevant. What was relevant is this NTRK3 gene fusion here, which is targetable. We identify this, and the clinician went ahead with the recommendation to treat with entrectinib. And as you can see here, that was a metastatic case. So we see 3 metastases, which really reduced after a few months of treatment. So that is really a test. The CMTA product is, I could say, universal, because we can use it for any type of tumor. And the timeline of our product, our company, I could give a short glimpse very shortly. We started the company with a strategic investment of 2.5 million from Kiagen, and started to develop the CMTA product. We received the German Innovation Prize, and kept going with for quite a number of years with R&D grants from European Community and the German government for 15 million. And during that time, we performed, the product was further developed, optimized. We introduced the CMTA in the market, which partial insurance reimbursement for some entities, and introducing in other countries now. One very important point, which is one milestone, is ISO 15189 medical lab accreditation for the CMTA. And we are now at a stage where we we are seeking for serious funding to ramp up our capacities and develop further in the marketing of this product. So who is Alacris team leadership? We are basically 4 main people, Hans Lehrer, George Church, myself, and Bodo Lange, who have long expertise in pioneering genomic technologies, which allowed us to develop the CMTA and the predictive modeling of drug response. And on this, I think that I can stop here and take questions. Thank you very much for listening.
Wonderful. Thank you so much for being here tonight. We're excited to have you for sure. And I know this technology, so from what I understand of your company, this technology is not new technology. It sounds like these technologies are all available. So one question I would have is, what's to prevent someone else from doing the same thing? And tell us a little bit more again about what really differentiates you from your competitors.
Yes, that's true. The technology is out there. We published those protocols and patents them long time ago. What prevents others to use it is the experience. We have been pioneered in these RNA-seq technologies. So we started ahead, we had a head start and spent a lot of time in this analysis. The reason is that other people don't use it is that the world has been and is still dominated by the DNA test because it's easier, it sells well as well, it still sells well. And a number of companies were not really in the swing of things to get going with the analysis of RNA-seq. So it's a lot of experience, of know-how. It's there. I totally agree with you, but it takes a long time experience to know how to deal with it, to be able to have a quantitative measure and to be able to turn it into a diagnostics, because you have to be sure of what you are doing there. So that's why we are very happy that we got the medical accreditation for this combined test using the RNA. So we are ahead of our competitors, even the large companies, because of this know-how, because we have learned how to interpret the data and to analyze it, yes, with the right tool. There's the analysis and the filtering of the events. So this is simply know-how.
That's fantastic. And before moving on to our questions from the audience, I noticed on your timeline you had something that was called the cup test. I'm assuming that's maybe a future product. Can you tell us a little bit more about that?
Yes, the CUP test is also something that has been around. So CUP stands for cancer of unknown primaries. They are up to 5% in Europe and the US. In other countries like India, they're up to 25% of CUPs to be analyzed. And we have developed this CUP test to be able to give a name to the tumor. So as the cups have unknown primaries, so we don't know where they come from initially, from the pathology, from simply looking at the tissues. Sorry, at the tissue slides, but with the molecular analysis, we can place the tumor and determine where it comes from, which is closer to a, let's say, colon cancer or breast cancer, and we can also read the mutations from the RNA-seq read. So this is a product that we are launching now. We start with India, and we will distribute it as well in other countries.
So again, probably the ability to treat these cancers more effectively, I'm guessing, even though you may not know where the primary cancer was.
Yes, but with the analysis, you know, because with a gene expression signature, we are placing the cup, when we get a cup, to the closest known cancer, because we have a number of signature for known cancer entities, and then we match the cup to its closest neighbor, so that we can then crack the actual origin of this cup, and that changes the treatment also, because we can also read mutation and fusions and so on. So otherwise the cups are treated almost exclusively with chemotherapy, and that gives a chance to the cup to have targeted therapy when it is possible.
Sure. Dr. Macaluso is in our audience, and he's curious to know, can this test be used on all types of cancer or only certain types of cancer?
All types of cancer, because the CMTA is based on sequencing the tumor, getting the RNA and DNA informations, and listing the alteration we found and matching the drug. So any type of cancer can enter into the analysis.
Wonderful. And Dr. Desai, who is an oncology fellow at Mayo Clinic, has a question for us. How is your technology different from Tempus/Foundation, who also report RNA data, although for research purposes only, it sounds like. What is the depth of sequencing in terms of number of genes, etc.?
Yes. Yes, it is true that Tempus and others also propose partial analysis of RNA-seq. We analyze data from Tempus from some of our clients, so we know how the data are. They also have different protocols. So we analyze with our technology, we analyze all the genes. So we have the 20,000 genes, not all, or 25,000 human genes. Not all are cancer genes, but we have them all in the analysis. This is how we can read the microenvironment as well, which are the immune cells in and around the tumor. So what differentiates us from Tempus is sometimes Tempus and other companies use a technology for the RNA-seq, which is they use capture, and that basically kills the dynamic range. So it's much more difficult to measure the quantities of the molecule. So if you are not quantitative anymore, it's very difficult to say if a gene goes up and down. But the main difference of with all of our competitors is the analysis and the reporting. They do not report the tumor microenvironment in the details that we are reporting, in neither the change of gene expression. We also report splice variants and so on. They are not getting into that.
So it sounds like your analysis and your report is much more comprehensive than what your competitors are doing.
Yes, that's it, because they had a different strategy, right? We have developed that over years. You saw on the timeline, it's a 10-year story. We were also part of the TCGA, ICGC for Europe enterprise. So we had a long learning curve as well, how to tweak, not to tweak, but to get the information out of this data. So that's really the main difference. It's much more comprehensive, it's more quantitative, and the report is more informative.
Wonderful. And you're in market like some of these others, which is fantastic. Well, Marie-Laure, thank you so much for being with us tonight. We appreciate you sharing your company with us. I think it's very exciting in the world of oncology treatment. A lot more cancer patients probably, again, are going to have successful treatments just by identifying those additional targets that you're able to identify. So thank you for all the work that you do.
Thank you.
All right, so next we have our second presenter of the evening, and that is Charu Singhal, and she is the co-founder and CEO of a company called Etta.
Hi, Katie. Thank you for inviting me here to pitch. Shall I share my screen?
You absolutely can.
Perfect. Hi, everyone. I'm Charu, the CEO of Etta. We're an AI company that specializes in building visual technology that can help detect tumors that are challenging to see with the naked eye. Thanks so much for inviting me back to pitch again. The last time I was here, I pitched our Etta dermatology tool in the Dermatology Pitch Club. Today, I'm pitching a new product for a specialty that we've expanded to. White light cystoscopy is used to detect cancers in the bladder. One of the most challenging cancers to detect is carcinoma in situ, also known as CIS. This is a non-muscle invasive bladder cancer that is challenging to see for even specialists due to the fact that there may be little to no contrast between the tumor and the surrounding areas of the bladder. Non-muscle invasive bladder cancers are cancers found on the tissue that line the inner surface of the bladder. 75% of bladder cancers are non-muscle invasive, and half of the patients treated with transurethral resection of a bladder tumor have a recurrence of the disease. While CIS is challenging to identify, the identification of CIS in a timely manner can have a major impact on follow-up clinical decision-making, including surveillance, cystoscopy, post-BCG treatment, and XRT patient selection. Currently, in order to detect CIS, there is a method, tool, and process called blue light cystoscopy. The company that builds the tool for this is called Cysview. This is a tool that has a high startup cost and a formula that must be rendered into the patient's bladder that can take 45 minutes to absorb. Not many hospitals utilize blue light cystoscopy due to the low reimbursement channels, the high cost to start up, and patient hesitation. At this time, there is no routine screening in the general public for bladder cancer for individuals that are at high risk for bladder cancer. A tool like this could provide support to a specialist at a diagnostic level and make screening more likely to provide early, accurate information. We are building a solution that overlays our AI real-time identification technology into the white light cystoscopy process to augment the intelligence of urologists by helping them identify CIS tumors. Here's how it works. As you can see, we're in the OR, and urologist here has inserted a stethoscope into the patient and is examining the bladder for anomalies. Here, our Etta tool has detected that there is a tumor inside this patient's bladder, and it is matching closely with CIS, as seen in our database through the models it is trained with. The urologist will likely perform a biopsy at this point. Although most hospitals do not have blue light cystoscopy available, all that conduct bladder cancer examinations have access to white light cystoscopy. As a company that values healthcare access and equity, we built a tool that provides the same ultimate utility as blue light cystoscopy, but simply uses AI overlaid into the white light cystoscopy that clinics already have. This results in no change to the existing workflow, no time added to a patient visit, and no discomfort for the patient. We started our company 3 years ago and have been achieving traction through a dermatology platform that helps clinicians identify, score, track, and map rashes and lesions. We found that some of the work that we did in scoring atopic dermatitis allowed clinicians to distinguish between texture and low contrast differentiations and nuances of depth within a lesion. We decided to apply similar methodologies to CIS in the bladder, and we're finding meaningful results in our early stages of testing. We layered a depth model with an edge detection model and an object detection model. The depth model differentiates the layers and textures within an image by comparing frames with their respective distances from the camera and the scope. This assesses the spatial structure of the individual's bladder from the vantage point of the scope, which can help a tool identify subtle distinctions in the contrast of the frames. We then layer it with an edge detection model to assess the texture and textural differences that could be shapes and anomalies that are related to CIS tumors. Finally, we have our object detection model that is trained on biopsy-verified CIS frames and from within bladders and white light cystoscopy. All of these models layered together build a tool that is an AI performing in real time more methods than the naked eye is able to perform. With just 100 CIS images and around 500 frames of normal bladders, we are able to achieve 88.9% precision, 72.3% recall sensitivity, and an F-score of 79.7%. In cancer diagnostics within data science, these are the metrics that are considered relevant when validating a model for diagnostic purposes due to the high risk of the nature of a model missing a cancerous tumor. Precision tells us how many of the cancer-tagged outcomes were actually containing CIS tumors, and recall, which is also referred to as sensitivity, tells us out of how many frames were given, how many of the frames that had cancerous aspects were accurately tagged as having those cancerous aspects. The F1 is considered the harmonic mean and an overall measure of how accurate a model performs. Given the extremely low amount of data that we started with, these results are very promising, and we are certain that with a bit more data and training and testing, we would achieve a very high level of identification that could truly change the landscape of CIS and how quickly it is managed. As we mentioned before, we have an existing AI dermatology platform that has achieved significant traction in the marketplace for the last 3 years. We last pitched our full-body real-time skin cancer product that is currently in pilot study at a research hospital based in Colorado. We wanted to expand our imaging technology in a place where it could be comparingly meaningful, and thus CIS was found to be similarly challenging in terms of visual identification. To roll this out, we are in the process of partnering with health systems that can collect more footage from the operating room, and we will use that to load up a clinical trial version of our technology. We'll conduct a trial based on the ethical guidelines from the UKCE, and we'll partner with a cytoscope company to scale up via a hardware pathway. And then we would like to see EdAssist utilized in every cystoscopy procedure. None of this would be possible without our amazing team of experts. We've always been backed by some of the most brilliant dermatologists in the country who helped us start and build some of the principles around our visual technology. We're now joined by Dr. Nitin Yaram, a urologist at a research hospital, who is helping us validate and bring this vision to life. We are so excited to make CIS tumors easy to identify in clinic, and I look forward to your questions. Thank you.
Great. Sorry about that. I was trying to put myself back on stage, but I think Mike got me taken care of, which is fantastic. Thank you so much for coming back and presenting on Etta again. I'm really impressed, by the way, since the last time you were here, with the traction that you have really been able to garner with the Etta dermatology product. Tell us more about that, and then what made you think that we could take this technology and apply it to bladder cancer?
Yeah, absolutely. So, we've been around with Etta for about 3 years. We had a whole, we're, as a founding team, we're engineers. So we were product people who teamed up with dermatologists. And we got to have like a really kind of fun pathway to finding product market fit, where we started off in a consumer space, helping people identify their own skin cancers, and then we took a telehealth angle where we were like the mediary between, seeing something on your skin and then connecting with a dermatologist, and then we took it to the ER with rashes, and then we took a pharma angle. So it really has been, dermatology was so much fun, to start up. And then in that process of evolving our product, learning the channels for how to validate a product, the correct way in the medical technology space, and how we were able to do that with dermatology, we felt like we were in like a good, safe space to kind of expand into other specialties and really do what we do best, which is visual identification.
So to clarify, and I'm just pointing this out because there's a couple questions in the chat about fitting into the clinical workflow. But with Etta dermatology, that was not simply, but software that could be used on a tablet, right? And in the office, in the emergency department, or at home to be able to do a skin scan, etc. When we're talking about the bladder cancer evaluation, go talk a little bit more about the clinical workflow and how you talked about partnering with hardware producers. Talk a little bit more about how you're going to integrate that into the clinical workflow.
Yeah. So as a company, we're really excited about sort of innovation being, less is more, where you use less expensive products, but you build software into existing tools. I think, in this kind of a world that we live in, that is how we can achieve something close to health equity, is by, doing scalable things like building technology that can really provide support. So, when we talk about partnering with healthcare or hardware companies, that would be like cytoscope companies to embed our software into their existing platform. And when it comes to the workflow, we know how hard adoption is in clinic, and putting in, another piece of hardware, especially if it requires a drug injection, education, patient, if you have to convince a patient to do it, there's so many barriers to entry that we think just, embedding software into an existing tool and partnering with that tool is just, a much faster, more clear-cut way to scale.
Well, and I can tell you on behalf of many of the physicians that are here tonight, putting a new technology into the existing clinical workflow is absolutely the way to go. And I know you know that too. A couple more questions from our audience. So with the precision at 88% right now, biopsies clearly will still be needed, which you mentioned in your talk. How do you propose to improve the identification? Could you get to a point where you don't need biopsies or other major interventions for your patients?
Yeah, absolutely. I think as it stands right now, you eventually do need to do a biopsy. What we could do is limit the number of unnecessary biopsies. I think that is a realistic target for us. Changing the workflow on the end where they're not conducting a biopsy, that might be a more difficult sort of pathway for us. But reducing unnecessary biopsies is a big one. Knowing where the cancer is, is our current goal, is to help them see the CIS tumors that you are very difficult to see. But after that, it would be reducing unnecessary biopsies. And then, maybe long term there will be, a better. But for now, I would say the standard is to always do a biopsy eventually.
Yeah, and I think my understanding is you're looking to partner with some urologists or uro-oncologists, really, because the more you feed your algorithms, the better that precision is going to get, right? So more and more studies where there is CIS involved, the better it's going to get, because of course it's AI. Is that right?
Yeah, absolutely. Since we are just starting out with this expansion, we do need more data. So any urologists that are interested in being data partners, any hospitals that are interested in doing that, the more we have, the faster we can, take something like this into market.
Awesome. And a couple more questions from our audience. One, are you able to accurately detect margins and be able to help guide definitive treatment, if that's the case?
We're not focusing on that just yet. That is kind of a next steps. We're starting with just CIS, and then, we're going to validate that and then continue from there.
Awesome. And I just have one more question, because I know you talked in the beginning about, and Mike actually put this in the Q&A as well, about the blue light cystoscopy. So we know that there is an alternative, a viable alternative out there right now. How is what you're doing an improvement over that?
Yeah, it's the cost to have it is around $116,000 for the hospital, and then there's like a $5,000 cost ahead of that. It's not reimbursable by most insurance, including Medicare. So that is like $3,000 out of pocket for the patient. It's very expensive, and I think it was like a great pioneer for CIS tumor detection, and kind of led the way into showing us that, like, hey, this is needed. I think that also, like the drug injection, there's so many barriers to hospitals and health systems with blue light cystoscopy, but it is great. We just see it as kind of like the first step, and then we're like the next stepping stone where it's like you don't need to do all of that. You can just use software overlaid into your existing hardware.
Wonderful. Well, Charu, thank you so much for being here tonight. I know there's still a couple of questions in Q&A, but guess what? Charu and Mari-Laure are going to join us for networking after we finish up here. And you, I hope all of you and our audience will as well, because they are ready to take more of your questions, which is really exciting. Thank you again for being here tonight. I'm going to say goodbye, and we hope you will join us in our networking lounge. Thank you so much for attending, and we hope to see you in another 2 weeks.